Hello students, today we are going to discuss on the Mendel's law of inheritance. But students, in previous lecture there will be some due to some technical problem. There was the topic. It means that the topic from the terms used by Mendel is skipped, and now after completing this topic we are going to discuss about the Mendel's law of inheritance. So students, first let's see the ninth term, that is the test cross. Students, what is test cross? The test cross may be defined as the kind of process of cross-hybridization in which the process of cross-hybridization is made between the individuals of F1 generation. Yes, students, when the process of cross-hybridization is carried between the individuals of F1 generation, such a type of cross-hybridization is known as test cross. Let's see on with the help of this example. Here I am going to take the hybrid tall plants of F1 generation. F1 generation. So select the selecting alleles are capital T small d and capital T small d. These are the individuals of F1 generation. Now, when the process of cross hybridization is carried, the capital T, capital T small d, capital T small d and small d individuals are obtained in F2 generation as a resultant. So students, what is the ratio obtained in this generation or in this type of cross hybridization? The ratio is obtained. This is the pure term. These both two individuals are hybrid term, and this is the pure dot, or you can also call it dot. Okay students, the pure dot, hybrid dot and dot. What is the genotypical ratio? The ratio of the result. Then we find that the pure tau ratio, hybrid tau, ratio dot. So students, the 1 ratio, 2 ratio, 1 is obtained. It means 1 ratio, 2 ratio, 1 is obtained. Genotypical ratio, it means 25% of the species become of pure breed type and rest of the dominance type, and rest of the 25% of the species also become homozygous type. It means the pure type, that is the recessive with the recessive character, and rest of the 50% of the species obtained is of hybrid type. So students, the ratio obtained can be also examined in the percentage 25%, 50% and 25%. But students, when we are going to study the phenotypically ratio, then the pure tau and hybrid tau both are taken together and the phenotypically ratio will be changed. Phenotypically ratio will be only given as tall ratio dwarf. Okay, no pure tau, no hybrid tau, only tall and dwarf. Then the result is obtained is 3:1. Okay students, this is the phenotypically ratio because the pure tau and hybrid tau, these plants appears tall in length or tall in height. So students, the phenotypically can be taken as the 3:1. Now students, we are going to discuss the Mendel's law of experiment. Mendel's laws of inheritance. On the basis of his experiment, the results obtained and the conclusion he obtained from his experiment, he gave the following 3 laws of inheritance. What are these? The first one is laws of dominance. Laws of dominance. Yes student, this is the law of dominance or law of dominance. The second one is law of segregation or purity of gamete. Law of segregation on purity of gamete. And the third one is law of independent assortment. Law of independent assortment. Okay students, these are the 3 laws given by Mendel by his experiments. So we are going to discuss these laws of inheritance one by one. First, I am going to discuss the law of dominance. Students, what is law of dominance? This law states that when a pair of contrasting characteristics is carried on the process of cross hybridization, then in F1 generation, only one character or only one trait is come in appearance. And students, the trait that comes in appearance, that is called the dominance, while the other which cannot comes in appearance, that is called the recessive trait. Yes students, the another is called recessive, that cannot be phenotypically come in appearance. So students, This is called the law of dominance. It can be understood with the help of an example. When the process of cross hybridization is made between the by selecting a pair of contrasting allele of the plant. So here I am taking a pure tall and a dwarf, or you can say pure dwarf plant of parental generation. Okay students, now here I am selecting their traits RID, the capital T for pure dwarf and pure dwarf small t. In Yapon generation, we obtain the result capital T small t, capital T small t, capital T small t, capital T small t. Okay students, in Yapon generation, this is the first failure generation. What we obtained? We obtained all the plants are hybrid crop. All plants are hybrid crop. Yes, students, they phenotypically are tall, but genotypically they are hybrid tall, because the capital T, it means the pure dwarf is the dominant character. It overlaps the recessive allele that comes in the pair of this individual plant. So capital T is the dominant trait, and it comes in appearance genotypically as well as you can say that phenotypically. But genotypically these plants are said to be hybrid plants. Okay students, because one of the allele present in them is of recessive type but does not come in appearance, so all the plants are visible as the tall plant. Okay students, this is the law of dominance, because only the dominant character comes in appearance or can be physically visible. Now students, we are going to discuss the second law. This is the law of saturation. Law of saturation or purity of gamete. Purity of gamete. Okay students, law of saturation or purity of gamete. What is this law mean for? Students, according to Mendel, this law states that the kinds of individual plants that are carried for the process of cross hybridization, there may contain more than one pair of dominant and recessive contrasting alleles. Yes students, and the alleles from the dominant or recessive traits, only one trait is going in appearance. It means only one of the traits come in appearance in the filial generations, because each of the traits are haploid in structure. This is hence said to be law of purity of gamete, and students, it can be understood with the help of the dihybrid cross example. Yes, students, the example of dihybrid cross. What is dihybrid cross, students? You all studied about dihybrid cross in previous lecture. When two pair of contrasting allele are considering for the process of cross hybridization, this is called dihybrid cross. And what the ratio you obtain? The 9 ratio, 3 ratio, 3 ratio, 1 is obtained in dihybrid cross. This is the genotyping ratio. Okay, students, what type of ratio? Genotyping ratio among the individual plant. Now, students, let's going on the how the dihybrid cross. Here I am going to select two traits. The one is stem length or height, stem length or height, and another is the flower color. What is, student? Flower color. Okay. Then pure tall or tall, pure tall, the capital T, and pure dot, small t taken as the contrasting allele, and here flower color red as dominant, capital R, and white, the small r taken as contrasting allele. Now, students, when the process of cross hybridization firstly carried, then the genes are, you can say that pair of alleles so obtained are capital T, capital R, capital T, small t, capital R, and small t, small r. This is the individuals of F1 generation. Now, students, the dihybrid cross is understood by the help of Punnett chart. Punnett was the biologist who gave a multiplication box for the process of the representation of the process of cross hybridization, and this type of multiplication box is after him said to be Punnett chart. What is, student? Punnett chart, because Punnett was the first biologist who gave the idea for the process of cross multiplication for this. And here I mix the total number of 16 boxes, because, students, 9 ratio, 3 ratio, 3 ratio 1 indicates the total number of 16. So, students, the first allele I take capital T, capital R, capital T, small r, the small t, capital R, and small t, small r. Here continue also capital with capital T, capital R, capital T, small r, small t, capital R, and small t, small r. Now, students, we multiply both of the contrasting pair of alleles, not mathematically but biologically. Okay. So here I am going to multiply the capital T, capital R, not T square, not R square. Okay. And now here the capital T, capital R, small r. The resultant traits are capital T, small t, and capital R, capital T, small t, capital R, small r. Here also capital T, This is the capital R and this is the small r. Capital D, small r, capital D, small d, capital R and small r. Here also capital D, small r, small r. Now students, the capital D, small d, capital R, capital D, small d, capital R, small r. And next individual is the small d, capital R. Also small d, and this is the capital R, small. The capital D, small d, capital R, small r. Capital D, small d, and small r, small r. Small d, capital R, small r. And this is the last pure recessive gamete. This is the small d, and small r, small r. So students, now we are going to nomenclate these plants. This is the tall red, also tall red, because capital R is the dominant trait and small is the recessive that overlaps by the dominant trait. This is also tall red, the tall red, tall white, tall red, tall white. This also tall red, the dwarf. Yes students, because both of the allele is of recessive type, hence dwarf and red. This also dwarf and red. This will be tall and red, tall and white. This will be dwarf, red, and this is also dwarf, white. This is the single individual so obtained that is having the characteristics of the recessive one with dwarf and white. Now students, we are going to calculate the genotyping ratio. Okay. Genotypically ratio. Tall red ratio tall white ratio dwarf red ratio dwarf white. Now students, counting the alleles having the similar characteristics, tall red 1, 2, 3, 4, 5. This is the 6, 7, 8, and this is the 9. So students, here I will write 9. After that, tall white. This is the first tall white, the second tall white, and that is the third tall white. So here I will write ratio 3. Now dwarf red. This is the dwarf red, and this will also dwarf red. So students, how many number of dwarf red individuals are found in the multiplication box? 3. And this is the only having recessive trait, that is the dark dwarf white, and this is the single. So students, the diagonal cross ratio 9:3:1 is obtained genotypically. Okay students, and we are discussing about the third law of inheritance in next video lecture, that is the law of independent assortment. Thanks students for joining today's lecture.